You recall that in bacteria, it's often the case that there is more than one coding region under the control of a single promoter. This is called polycistronic messenger RNA. So for example, here we have a messenger RNA molecule, 5' to 3', that has a leader region followed by two protein coding regions, one here and one here. And between them there's a spacer region, which can in some cases be very short, from -1 to 40 bases between them. Obviously -1 means that the stop codon of the first coding region overlaps the Shine-Dalgarno sequence, or in some cases even the initiation codon of the second. And I'll talk about that more in just a minute. So let's assume that we have a spacer region of some reasonable length. It can actually often be even more than 40 bases. The translation initiation region includes the Shine-Dalgarno sequence, which you remember is where the ribosome binds, and the initiation codon, which you recall is where the first amino acid is inserted using the initiation FMET tRNA. That's followed by a coding region shown here with the slash marks, and then ends with the stop codon. So this is the messenger RNA. This is the sequence that binds to the ribosome, the Shine-Dalgarno sequence, the initiation codon in AUG, and the stop codon. That's followed by the second translation initiation region, which has its own Shine-Dalgarno sequence, its own initiation codon, its own stop codon. In between is the coding region for this gene. While only two are shown here, it's possible to have 3, 4, 5, even as many as a dozen genes, all under the control of the same promoter on the same transcript. So the way this works is that the Shine-Dalgarno sequence and the initiation codon are the binding sites for the ribosome, and the stop codon is the region where the ribosome dissociates. Each translation initiation region can bind its own ribosome. So this could be ribosome 1, this can be ribosome 2, and if there were other open reading frames or genes downstream, each one of those binds its own ribosome. Each of these ribosomes that binds then makes a polypeptide that corresponds to the gene that is encoded in this region. I mentioned that in the case of a -1 intercistronic region, you can actually have an overlap between a stop codon and a start codon. In that case, there is no Shine-Dalgarno region because the ribosome starts here, and rather than terminating at the stop codon, it simply shifts by one nucleotide and initiates a new protein right here at this initiation codon. This situation is relatively rare, but happens enough to suggest that allowing a single ribosome to continue to the end and then hop by one nucleotide and reinitiate is not unusual. Often when you see this, these are proteins that have to work together. So for example, protein 1 and protein 2 make two distinct polypeptides. However, those proteins are two subunits of the same enzyme. And so it's possible that by organizing these genes such that the stop codon and the initiation codon overlap by a single nucleotide, it ensures that those two proteins are made in equal amounts, which is what you would need if they were subunits of the same protein. I want to talk for a minute about translational coupling. Now first I want to mention that translational coupling is not the same thing as transcriptional translational coupling. So transcriptional translational coupling says that translation happens immediately after a transcript is made, and that a transcript and a protein are made almost the same time by ribosomes that bind immediately to the transcript. Translational coupling is something else. Definition of it is that translation of an upstream gene, for example gene A here, is essential or is required for the translation of the gene that's immediately downstream. So we have two genes, one transcript. Each gene has its own Shine-Dalgarno sequence, its own initiation codon, its own stop codon. However, the structure of this region is such that the initiation codon of gene B, right here, is embedded in a hairpin that's close to the end of gene A. So how this works, if we're looking at the structure of the messenger RNA, is that we have gene A, we have in green the stop codon for gene A, we continue through the region between the two genes in the messenger RNA, and we come to the initiation codon. Now, because of the secondary structure that comes from the sequence of the region from the stop codon to the initiation codon, the result is that this initiation codon of gene B, shown here in blue, is in a hairpin structure. That means that it's difficult for a ribosome to initiate there unless there is a ribosome that translates through to the end of A. So a ribosome binds at the Shine-Dalgarno sequence and the initiation codon goes through to the end. And as the ribosome moves through the secondary structure to the stop codon, it disrupts the binding caused by the homology between these bases, disrupts that secondary structure, thus opens up this codon and makes initiation of gene B accessible. So in this case, if a ribosome stops for some reason before the end of gene A, you do not get translation of gene B. Note that this is not transcriptional, it's entirely translational. So there is a transcript that goes all the way from the 5' to the 3'. The transcript is there, but the initiation codon in this transcript is blocked. As a ribosome moves through gene A, it opens up the secondary structure, allowing translation of gene B.